everyone. Well, it's 11 o'clock here in Bloomington, Minnesota, so we're going to get started. Thanks, everyone, for joining our webinar today. My name is Jackie Carville. I'm the Marketing Director here at Immunochemistry Technologies, and I will be moderating this webinar. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. Simply type your question into the chat dialog at the right side of your screen and select Send to Host. If you have any technical difficulties during the webinar, please send your chat to me and I will do my best to get you up and running. We will be posting the video recording of this webinar within a day or so afterwards, so look out for an email from me after the webinar with a link to this recording. Now I'd like to go ahead and introduce our speaker, Dr. Brian Lee. Brian is the president of ICT and leads our R&D team in addition to providing excellent technical support to our customers. Today he will be presenting some methods in order to simplify your ELISA development process. And with that, I'm going to go ahead and turn it over to Brian.
Thanks for the intro, Jackie. Given the type of products that we sell at Immunochemistry Technologies, we thought it would be a good idea to maybe provide some insight into ELISA development, since a good share of our products relate to building your own ELISA. The ELISA as a assay is typically considered to be specific, low-tech, sensitive, and quantitative. In many cases, it is the go-to method because of this for assessment of various scientific, biologically significant, commercial, environmental, what have you, analyte targets in plasma, serum, environmental samples, whatever. And so that's the basis for this little brief webinar here today. ICT, Immunochemistry Technologies, which is why we say ICT, is a small company in Bloomington, Minnesota, and we were founded in 1994. And in that initial services company, we were concerned with helping people do conjugations and develop their own assays and various things like that. So we were kind of a service company, hands-on service company. And then in 1997, we said, well, hey, we got all these products that we have, well, all these assay-type components that we could convert to products. And so we started selling what we call the ELISA Solutions projects. And in 99, we decided to branch out into fluorescence-based assays, because that was a growing area with advancement in fluorescent probes and the type of fluorescence reader, plate reader technology. So we also do that on the other side of our company. Now, the present focus on today's products is on cell status. Pardon me. The present focus for today's assay would be on the ELISA. And just to give you a little background as to our qualifications, we have extensive experience in protein chemistry, antibody purification, protein conjugation. The founders had a lot of prior background history and multiple different types of enzyme EIA immunoassay development formats, like agarose, immunoelectrophoresis. Some of that's going way back if you're older. Western blotting, lateral flow, that would be your rapid test format. Think of your pregnancy test or some other test where you need a result very quickly. Homogeneous enzyme immunoassay format, that's the type of assay where you throw everything into a tube and then you basically don't have to do anything until you're ready to add the substrate. And that is very interesting. It's very complex, but that is not what we're going to be focusing on today. For today's, we're basically focusing on the ELISA topic. So we sell or offer a bunch of ELISA-related products. And the whole concept here is that we are promoting the build-your-own theme products type concept, where you don't have to try to figure these things out on your own. You can take a look or check us out and see what products we have that will help you develop your ELISA, whether it be an antigen down type ELISA for screening antibody and serums, or whether it be a sandwich assay like we're talking about today. We offer these products already made. We know they work, and we felt that it was worth selling them to other people who really haven't had that much experience doing this type of thing. And in that regard, we offer a starter antibody-sandwich or antigen-down ELISA with 2 different products. And what they do is they kind of walk you through the steps of developing your antibody sandwich or antigen-down assay, and they provide you the proper media-type solutions to actually get started. Now, they're not going to be perfect. You'll probably have to optimize or modify, but at least get you started. And once you can get started, then you can know, what do I have to do next? It's more sensitive, or I got too much background. There's various things you'd be looking at. All right. Everyone's seen one of these in Immunology 101. And essentially you have your IgG, and we see Ig because some people use other types of antibody isotypes for the ELISA. But in general, it's an IgG, and it has its basic antigen hypervariable antigen binding region at the top, and then within that region, then you have a what we call a paratope. You don't hear that too much anymore, but it's a small, maybe 15, 20, 25 amino acid size region where the antibody and the antigen, the target, make intimate contact, and that's where it binds and stays there. And the hypervariable region consists of both the heavy chain and the light chain. And then, of course, the bottom end of the antibody here is the FC portion. Stands for fragment crystallization. Again, that's old term, but that part of the antibody is very important too, because it binds to FC receptors on B cells, natural killer cells, macrophages, neutrophils, the whole list, and that's used for immunomodulation type activity. So it isn't just a one-ended molecule like you might have thought when you first started taking immunology. But it is the key discriminator, if you want to call it that, for how we make these immunoassays work. You have to have something that is very specific. You have to have something that grabs hold of something and hangs on to it so that you come in and then measure it. And if you didn't have a specific or high affinity antibody, you couldn't do any of these assays. It doesn't matter what type you're looking at. Now, on this slide, we're looking at mono and polyclonal. You guys all know what they are. Monos have some advantages. You get uniform specificity, uniform affinity, and this is nice. I mean, if you're really producing an assay, especially a commercial type assay, you want this type of thing. And if you don't let your liquid nitrogen tank dry out, like I've known people to do, not myself, but you'll have a reliable supply of monoclonal antibody in the future. The downside of it, of course, is, you can lose everything if you don't keep your tank filled up, or in some cases, you don't really have a mono. You have a diclonal, which case you might have another cell in there that's out competing the good guys. So there are some downsides to the monoclonal. They don't like being conjugated as well. There's just one molecule versus a whole bunch. So you have a higher probability if you're using a monoclonal in your assay, and you're relying on that. If your HRP conjugation technique isn't very good, you might do something where you denature or interfere with the actual ability of the sandwich antibody, up antibody to bind. Then there's polys, this one on the right. And from my experience, polys are less expensive to generate large quantities of antibody. It doesn't take quite as much time to get them going. Polys give you the opportunity to recognize a bunch of different sites. So you're not just stuck on one site or a redundant type site like a monoclonal would be looking for, but you can look for all sorts of different epitopic regions on your target analyte. Because they're polys, if you're going to do any modification to them, there's a lower probability that you're going to have a population-wide affinity or activity hit when you don't do your conjugation right. So in the case of polys, however, they do require affinity purification, and because you are using a large pool of IgG, that's good, but you have to make sure that you have a large enough pool so when you run out, you have to start new animals and start new immunizations. So there is a downside to polys, but if you got a good freezer and you store them correctly, they're every bit as good as monos, other than for the fact that if you're going to use a sandwich assay, you might want to use a monoclonal on the plate as a capture antibody, because it is going to be the most specific and less likely to cross-react with some other analyte, non-analyte in your sample. So the monoclonals give you the comfort zone of being very specific. All right, this is an example of different ELISA formats. You've got the antigen down on the left, and this is a little unusual because you usually don't put antigen on the plate and then come down with a signal-generating antibody on top to detect it. But in some cases, you may want to do that because you want to find out, well, is this stuff I isolated, is it really what I think it is? And so you absorb it onto your plate, block it, and then come in with an antibody you know is going to detect this, and if it lights up the plate, well, then you're probably good. In the second case, you have an indirect antigen down ELISA, and this is typically how you run things for antigen down. You have your analyte on your plate, and then you incubate the plate with the host antibody and host serums containing antibody, and then you come in then with an anti-isotype. If it's human antibody, then you come in with say a good anti-human IgG if you're looking for that, and get your reading that way. And then, of course, the more antibody you had on there, the more secondary antibody conjugate you have. Therefore, you get more color, more product development, and they're not linear per se, but there is a dose response as far as the amount of antibody concentration that's present. In the case of a competitive assay here, the third one, this is kind of cool. You can actually, if you have an analyte that's kind of small, and you really can't put stuff on there because sometimes smaller molecules are harder to modify, you could do a competitive assay where you absorb the antigen or analyte to the plate, and then you compete with, say, inhibitor or a competing antigen. So basically, you're trying to measure how much antigen is present in your sample, how much analyte is present. You have a known amount on the plate, then you incubate with your sample, and those samples will contain various amounts of, you think, this other analyte, and we use an inhibitor, but it could be just your analyte you're looking for. And so the more that you have present in your sample, the less antibodies going to be able to bind to the plate because it's going to be tied up by the antibody that's in the, or tied up by the analyte that's present in the serum sample or whatever your sample is. On the far right, it's a capture sandwich ELISA. This is your typical format where you have a capture antibody absorbed on the plate, then you capture an analyte, and then you wash and come back in with your antibody conjugate with some type of signal generation. And this is classic antibody sandwich type format. Now, in many cases in research, and I have to say this because I'm trying to be fair here, in many cases researchers don't have a lot of antibody, and that up antibody they don't have very much of because it's hard to come by, or it's a mono or something. And so what they will have to do is they will have to instead come in with their up antibody, and then say their up antibody is a mouse antibody and the plate captures a rabbit. then they'll have to come in with an up antibody, mouse anti, whatever that analyte is, but it's not labeled, so you have to figure out how to get the signal out of it. And by doing so, you have to come back in then with a third antibody. So there's an extra layer here on this one on the far right, and the third antibody would be anti, say, mouse isotype to whatever the other side up antibody for the sandwich antibody is. So it's actually a 3-layer thing. It's a lot harder to do because you got like 3 layers instead of just 2. So trying to develop and optimize an assay like this is a lot more difficult than just having a up antibody that's directly conjugated with your enzyme signal generator. All right, in this slide, we mentioned a few ways that you can get false positive interferences, and there are a lot. Limited time today, so we just kind of grabbed a few common ones and ones that you might run into just for demonstration purposes. On the well on the far left, that person who was running that assay was a novice and decided that, or didn't know, that they had to block their plates after they coat with the antibody. So what happens is when you come in with your conjugate, which is the yellow structure there with the enzyme, the blue oval enzyme on top, that'll just bind to the plate. Basically, you don't have an assay; it's a mess, and then you'll hopefully someone will tell you, well, you're supposed to block that first, and then you'll quietly not tell your boss or your advisor that you didn't do that, and then the next time you'll come in and block it the proper way. Now, in the second well, you have nonspecific binding issues because you've got sometimes the matrix of the sample matrix, that's your serum or urine or whatever your sample matrix is, you can have materials in there that can non-specifically bind to your blocked plate surface. Just because you block something with a blocker does not mean that things aren't going to stick to it. It's less likely than sticking to that sticky polystyrene plate, but that's the tricky part of ELISA, especially sandwich ELISA assay development, is what kind of a blocker can I use that's like Teflon and basically nothing wants to stick to it. In this case, you're getting a false positive signal from the material stuck to the plate. Then you come in with your conjugate, it goes, well, here it is, and now you do not have an assay. In the third situation, you will occasionally have a capture antibody, and this is more rare, where it will actually bind to your signal generating antibody. This can happen. We just threw it in there. There's other cases where that signal generating antibody is not binding specifically, but it's actually just associating with the FC region of the capture antibody on the plate. It's a polysaccharide type phenomenon, and it's very frustrating because if you're using a rabbit antibody, for instance, a rabbit poly, you've got a rabbit poly on the plate, and then you come in with that same rabbit poly with say an HRP conjugate on there, they will want to stick to each other for no good reason, and it can really give you very high frustrating backgrounds unless you pre-absorb that up antibody, which I've had to do in several cases, to remove those guys so that the guys that are left in there will not non-specifically interact with the FC regions of the capture type antibody. And that's with rabbit. It may occur in other situations as well, but I know definitely rabbit can give you a problem. And then you've got the fourth situation here where you've got cross-reactivity with something that's in there, and in this case, it just simply competes with the other guy. But you typically in your sample matrix, you have a lot of whatever that is. So you might have a certain amount of analyte in there that's not very much, in the picogram per mil range. But if you have some cross-reactant, it's very possible that could be up in the nanogram per mil range, and it will, just get in there and just bind to everything, and it doesn't even care about your analyte because it's got all this other stuff that's kind of tie it up. So you're going to get a large positive signal that way, and you've got big problems because you've got something in there you didn't know about, and now you've got to figure out, well, am I going to have to get some new antibodies? You have to do something. Now, maybe you need a monoclonal, but it means you got to go back to the drawing board. And if you're a first timer on this, that isn't really what you want to be facing at this point. Okay, in this slide we have the false negative potentials, and again, not all conclusive, but you have a situation where you could have something actually in your conjugate diluent. Again, not necessary you would expect, but sometimes a conjugate diluent can bind to the conjugate, the say HRP IgG conjugate, and in so doing, it interferes with its ability to sandwich with the analyte that was captured by the antibody on the plate. In the second case, where you have a reaction where you've got basically the capture antibody sees something, and for some reason, again, maybe you got a mono there, for some reason the up antibody does not recognize it. So basically, when that happens, there's nothing for the up antibody to bind to, so you don't get much of a signal, even though there is analyte present. In the third case, this is probably a little bit more common than most people would like to realize. You have, for regulatory purposes, a lot of these analytes have what we call soluble receptors, and they're on there for a good reason, for regulatory purposes, and they bind to your target. It could be your cytokine or whatever it happens to be. Now, that's all well and good for regulation, but if you want to run your assay, your capture antibody sitting on the plate is going to have to compete with this already bound soluble receptor. And when it sits there, it blocks the epitopes on your target, and you essentially don't get the signal you should. So I ran into this at a prior job on a couple occasions where they didn't know there were soluble receptors, and lo and behold, there was, and then we had to get creative with our assay diluent to try to disrupt that, and you can do that. So just letting you know, this isn't the end of the world, but you can put stuff in there that because the soluble receptor has a lower binding affinity to your target analyte, if you disrupt that, you'll still have enough antibody capture activity to still grab the thing. So again, that's where ELISAs become projects. So I want to mention a couple other things on this too that we did not have room on here. You can also get human anti-mouse or human anti-other antigen, and that could actually bind and tie up the capture antibody in such a case that it doesn't function. So it can interfere sterically by coming in and binding to, say you got a mouse on the plate or some other animal, and the serum for whatever reason has antibody to it. That'll interfere in a negative way, and so you have to try all these things out. It's trial and error. You don't always know this, but this is why you spend time on these, and this is why you listen to simplified seminars like this. Okay. All right. We thought we'd just put in just a basic stereotype flow chart on how you do a antibody sandwich assay. You coat your plate, you better block it, and then you wash, and then you capture your sample, whatever that may be, or your standards. And then you come in with your secondary antibody, which sandwiches the captured antigen that's on the plate antibody, and then you wash again. You add an enzyme substrate, usually it's TMB if you're using peroxidase, and then you stop the reaction so it doesn't keep going, and you can actually get a stable reading on your plate reader. And this is just, basically sandwich assay 101. We just showed that just for fun for some of you who maybe weren't totally aware of how these things are done yet. Now, in this slide, we're going to talk about coating buffers. Traditional coating buffers started out back in the I'd say probably the mid to late 70s. And that's when I was cutting my teeth in this area. That tells you how old I am. And anyways, the plates that they put out there weren't very good. They did not do a very good job of binding proteins, and good luck if you had a LPS or carbohydrate or something, because that was really a problem. So in order to make things work, the folks said, okay, we're gonna modify our buffers to try to get our proteins to stick, absorb better to the plate. Now, these might be antigens, these could be your antibody. Regardless, the buffers were low ionic strength, meaning 0.03 to 0.5 molar carbonate. Usually the pH was pretty high, 9.6. The upside of this was that the low ionic strength helped the proteins or antigens or antibodies interact better hydrophobically with the plate surface, and the plate, for most polystyrene plates, you have a hydrophobic surface where they've come in after the fact and modified with radiation or some other technique to create charges on the plate. So you have not just a hydrophobic surface, but you also have a charged surface as well. So the low ionic strength was used to enhance hydrophobic absorption. The high pH was used to make sure that your antibody has a charge. Now, the isoelectric points of most antibodies is in the 6.8 to, some as high as 9, but most of them are in that neutral to mid-7 range. So when you adjust the buffer to a pH 9.6, all the proteins in there, the antibodies or antigens, are going to be in the negative charge form. The further up the pH scale you go, the more electron accepting you have, and so you're going to, all those NH3s are going to be now NH2s, and your overall net charge of the antibody is going to be negative. And so that'll help it stick to the plate. So that's the basis for the why I say the rather harsh conditions for the initial coating buffers. And I guess this slide, I'm just talking about so I skipped over this one. I was reading my notes, and this just reiterates what I just said. Now, we wanted to make a better buffer. If we're going to sell things commercially, it can't just be the the original, format. We wanted to improve it, and so the buffer that we sell, the plate coating buffers that we sell, have a slight alkaline pH, and they're around 8. And we don't try to use low ionic strength because it isn't necessary with today's high binding plate chemistry. You don't have to go to those extremes to get your protein or whatever to stick to the plate well surface. So our buffer is different than the other ones because we don't use a super high pH, and we don't try to, basically low ionic strength, essentially in some cases denature the antibody by incubating in a very low salt concentration. We use rather more isotonic type salt concentrations, so that after overnight incubation, you haven't done a real number on your whatever you're putting on there, whether that be an antibody or an antigen or whatever. If you damage it because of the incubation process, then what have you really accomplished? You did a great job of sticking something to the plate, but how effective is that something after being exposed to these harsh conditions overnight? So that's the take home from there. All right, I guess we're back to blocking again here. Blocking is not a surefire way to avoid all problems with nonspecific binding. In this example again here on the far left, the person just didn't block the plate, and obviously you've got problems there. And in the second case, you have the antibody the plate is blocked, but it's not blocked very well, and there's certain blockers that don't block very well, and so there's still space there for that signal generation antibody, the yellow antibody fragment there, to bind to that plate directly. In the third case, you could have a cross-reactivity with the actual blocker. That's very rare, but it can happen. And in the fourth situation on the far right, you can get nonspecific binding of sticky IgG conjugates to the actual plate well surface, and that's basically the thing on the yellow antibody on the right in that far right well. There are other situations where you're not having where you're actually having a media matrix-based, sample matrix-based analyte that's sticky, and it sticks to your block plate surface, and then your secondary antibody comes in and says, well, I recognize this, so now you've got nonspecific binding because it's seeing something it can actually react with that's stuck to your block plate surface. Now, there are a couple other ways to have problems with running ELISAs, and the nonspecific binding issue is a huge one. When some people have this type of IgM type form, and they're totally healthy, so I'm just telling you, but the IgG, or IgM I should say, is sticky carbohydrate, and it wants to stick to your blocked surface. It's very frustrating because you blocked the surface and you didn't block it so that IgM could stick to it, but it does. Now, IgM's stuck to your blocked surface during your sample incubation. You wash, do all that stuff. You come in with your signal readout antibody, and it just sticks to the IgM that's stuck to the plate. So when you look at your zeros, instead of zeros, you got a reading of like 3.0 or something, and it was because the signal generating antibody is not specifically binding to that IgM that decided it wanted to stick to your plate. Now, there are treatments for that, and one of our buffers actually addresses that, where we try to convince the sample IgM that's sticky to not stick to the plate surface. Again, encountered that in a prior job, and it took a long time to develop that assay because certain people had this IgM phenomenon, and we know it is because I actually did some experiments where I took a rabbit anti-human IgM and incubated it with the person's samples, and then the problem went away. So I know that it's IgM based, and that's as much as I can tell you at this point. All right, we have blocker stabilizers, and everybody else has blocker stabilizers. What's the difference? What are they supposed to do? All that. That's what the purpose of this slide is about. The idea is the blocker stabilizer is supposed to minimize sample matrix, like we were just talking about, sticky things from sticking to the block plate surface. In addition, sometimes you have conjugates that you didn't do very well, and those conjugates themselves are sticky, and you want to minimize the sticky conjugate from non-specifically binding to the blocked plate well surface. So in both cases, you have something binding to the block plate well surface, whether it be something from your sample matrix, or maybe it's your sticky conjugate, and these things do happen. How do I still use that conjugate without having to go back and redo it again? And these blocker stabilizers are supposed to address that fact. Now, our blocker stabilizers, we've tried very different additives in there, and we did that on purpose because in some cases a certain blocker type surface will work better in a situation than in another situation. There is no one size fits all blocker. So if someone's telling you that, somebody's blowing smoke and it probably isn't you. So just take that into consideration. There are different blockers, and there's different blockers for a good reason, and because there are situations where one type of blocker with one type of additive will work better than in another case where the other additive is added and it works fine. But sometimes it becomes kind of a treasure hunt to find which one works the best. But you have to at least try that out if you want good backgrounds in your assay. Now, the secondary objective to blocking the plate is to preserve the integrity of your antibody or your antigen. And it's very important that when you dry those plates down, that they still resemble what you started out with. If you have a coated antibody on there, it still acts like an antibody when you rehydrate the plate again. These blockers are made so that you can make a whole bunch of plates at one time, so they're all consistent as far as precision goes. Then you can block them all, dry them down, store them at 2 to 8 degrees C, and a year from now, they should still work. You come back in with your sample, and a year later, you open up your little pouch where you stored your plate with a desiccant, and you take that plate out. Yeah, I suppose the magnitude might be down a little bit, but that you are still going to get a functional assay from that blocked and stabilized antibody-coated plate. And that's a big difference if you think of what you'd have to do if you had to basically coat and block and then try to use that plate the same day. That makes for a real long day. And I know a lot of people, intro people in academia, who are just new to this, that's what they do. So, if you're a grad student, hey, well, you're supposed to be there 16 hours a day anyway, so, what is your time worth? But you get what I'm saying, okay? I did the grad school experience, too, so I can tell you that's kind of the attitude at times. So I think sample matrix challenges are going to be an issue, and we need to be able to compensate for these matrices, because you don't know what you're going to run into. You're not sampling everything in PBS. It's too bad, but our serums are serums. They're not made out of PBS, and there's proteins in there, and there's regulatory proteins, there's lipids, all sorts of fun stuff that can stick to your plate. And when it does, it can stick non-specifically to your conjugate, and then you have a mess because you can't get a nice low zero background. Now, this slide we're talking about sample and standards. We offer both a sample diluent and an assay diluent. And if you're not familiar with the vernacular, say, well, what's the difference? And I don't blame you. Well, the sample diluent is what it says. It's used to dilute out your sample, your serum, urine sample, plasma, cell culture media. It's used to dilute your sample out to the level at which you can detect it, in your assay. In some cases, you can dilute the sample way out and you don't have a matrix effect. If you can dilute your serum matrix out at least 5-6-fold, the matrix issues drop way off fast. But if you have to look at that on a basically neat or 1-to-1 or 1-in-2 type concentration, usually the matrix is big time, and the sample diluent at least lets you dilute that sample out in that process and gives you a stable buffer for that particular. So while the samples are sitting on your desk and before you pipetted them in, it'll give you a stable buffer to preserve the samples until you get them into the plate. Now, the other concept is we have the assay diluents. And the assay diluent is similar to the sample diluent, except the assay diluent is designed to try to equalize the complexity of the matrix in your standard curve wells versus the complexity that's in your serum samples, for instance. Now, why do we care about that? Well, if you run an assay in easy buffer like PBS, BSA, and you compare that to a spike sample that you ran in your serum matrix. Your easy sample, your permissive matrix, will capture very efficiently in your standard curve, but it will not capture very efficiently in the sample wells, and so you're going to underreport what's actually present in your sample wells. And this is the biggest frustration that I know of for doing ELISA, sandwich ELISA diluents. Diluents needed to, try to minimize this interaction. How do you make the two equal? And when you're just working in simple samples, even like cell culture, you say, this is a breeze. But as soon as you get a bunch of serum samples and you do your spikes in there, you know how much you put in there, and you know how much your standard curve says. And when you read your samples, serum samples, off the standard curve, instead of saying 100 picograms per mil, maybe it says 50. Now, they didn't go away. The problem is they just simply didn't bind to the capture antibody as efficiently because of the matrix components. Let's go down here to, where are we? I want to, real quickly here, just try to visually show you the matrix effect. We can't do 3D here and all that, but just for grins, we drew a picture of two wells. In the well on the left, this would be like your calibrator diluent. You got your standard curve in there. It's a breeze for the green analyte to make it down and interact with the capture antibody in the plate. Now, the well on the right, containing serum, there's a lot more stuff in the way. And I'm using this, there's other interactions, but just to show the point is a more complex matrix will make it harder for that top green analyte ball to make it down to that capture antibody. So the binding kinetics in that well are much slower than they are in the very permissive calibrator well. Well, that's what happens. You get 100 picograms per mil in the left well, and then in the right well, maybe you get 50. Well, that's a problem, and underreporting is something that people have to address when they're working with these complex matrices. So we show here how if you add an assay diluent to your plate wells, the assay diluent has to go in all wells that are going to contain samples, every well. You add the assay diluent first, and then you come in with your samples. And what the assay diluent does is it introduces these other interfering factors that will tend to foul up, complex up the calibrator standard to a greater percentage than they do the already complex sample matrix situation on the right. So the net effect is that it brings the binding affinity of the capture antibody on the left side down to the level of the way the poor capture antibody has to work in the serum sample on the right. So you don't underreport as much. Eventually, you try to match these up so that when you put in 50 picograms into your sample and you read it off your standard curve, you read somewhere between 45 and 55, picograms per mil off your standard curve, and not like 20 or something like that. Now, the issue goes beyond there. Trying to make this, hang on a second. All right, I got it. To show this graphically, if you had enough time in the day, your complicated matrix samples would eventually approximately catch up to where your non-complex, like say your calibrator diluent samples were. But we're showing this here just to show that within an hour's period of time, because of the capture efficiency differential, the capture efficiency of the capture antibody in the standard curve wells with a very nice, easy matrix is, some certain level. And comparatively speaking, that same amount of capture efficiency in your sample well is about half that, say after an hour. This is overblown. You probably wouldn't reach this much of maximum after an hour, but you get the point. Is that typically none of these ELISAs are run to equilibrium. You don't have enough time. It's not worth it. We try to capture a signal at a point where it's starting to flatten out. And so ELISA is not endpoint. It's not total equilibration. It's getting to a point where it's not changing too fast so you can get an accurate reading. And so what our assay diluents are trying to do is try to bring that yellow curve down closer to the blue curve so they're equally compromised. And in that case, when you get a reading, it's going to be similar in the standard curve well, you hope, as it is in your sample wells. Otherwise, you're not. You got apples and grapefruits. It's not even the same matrix. It just doesn't work. You don't have an assay. All right. Conjugate diluents. As you know, there's lots of conjugate diluents products on the market. And their job is supposedly to stabilize both the antibody component, as well as in most cases, HRP component. If one of those goes out, you don't have an assay. If you have a good antibody stuck on there, but the HRP is not stable, you don't have an assay because you don't have any signal. If the HRP stays stable, but the antibody gives out, you don't have an assay because you got nothing to capture. So you really need to stabilize both the structure of the IgG, the tertiary quaternary structure of the IgG, as well as of course, the functionality of the hypervariable region, which is all important, because if that goes south, even though the rest of it's okay, again, you don't have an assay. That antibody is non-functional. So the conjugate stabilizer needs to stabilize the hypervariable region of the antibody so they can continue to bind to the target analyte. Now, in the next slide here, we show that we also have to worry about the happiness of the HRP integrity when something is stored for a longer period of time. Now, these conjugate stabilizers are meant so you can make something up to be used at a 1x level. You don't have to. The end user doesn't have to dilute it out. They can just dump it in and use it as is. Now, when you do that, you have to make sure that both your HRP component and your antibody components remain stable over time. And the idea here is we're showing that we want to also stabilize the HRP tertiary structure, and with special emphasis on the porphyrin ring, which is where the redox reaction occurs in HRP, and electrons cycle around, and those happy faces are just our way of easily saying to you that we are stabilizing both the porphyrin ring as well as the tertiary structure of the actual protein portion of the HRP. Now, our last slide, or almost our last slide, I guess we're reiterating the fact that we're really in the business of offering people a start to finish type product line. So you can purchase from us, if you wish, everything to basically start out your assay, coat your plate, block it, dilute out samples, wash it. We sell wash buffer. We sell all liquid components that it takes for you to develop your assay, including the substrate and the TMB substrate and the stop solution. Our products have been basically, we have a worldwide distribution of these products, and they've been highly cited in numerous peer-reviewed journals. So other people have used it. They like it. So essentially, we have a satisfied customer base, and that's good because if people remember when things don't work a lot more than when they do, and if if you ever get into this business, you'll realize that yourself. Our company mission is to always provide excellent technical support both before and after the sale event. When people will call me up, I will answer questions. I don't know if they're going to buy anything, and I don't care. I want to be a source where they can call me and they can get a straight, honest answer, an accurate answer for what they need to do. Now, if they call back, that's great. If they could use our stuff, that's great. I would rather have people see us, our company, as a source of integrity and good products. So that's our sort of our mission statement there. All right. And the most important slide. Well, this is our team, and you see we're a huge company. There's many of us, and this is a group that's responsible for managing our day-to-day business operations. And essentially, most companies are really nothing were it not for the employees who make things happen. They aren't just products. They aren't just a bunch of documents and bills and to-do lists. They're people. Without people, good people, the company's nothing. And just to point out, I'm that bald kind of seasoned guy on the far right. So I'd like to thank everybody for tuning in to us today, and we'll take any questions. And if you have questions, if there's time, we will do what we can to check that out. So thank you, Jackie.
Thanks a lot, Brian. We have a lot of questions chatted in, so considering our time, we're only going to be able to address a handful of them, but please feel free to keep chatting in your questions. If we don't get to your question within the webinar here, we'll be following up with you right afterwards with a real detailed email going through that and ways for you to contact us to talk through that question. But first, Brian, we have somebody who's wondering why they can't just use the old non-fat dry milk blocker recipe to block their ELISA plates. It seems to work for them before when they were blocking nitrocellulose or PVDF-type membranes in their Western blots.
Wouldn't it be wonderful if that were the case? Stuff is cheap enough. In fact, the older the non-fat dried milk, the more off-brand it is, the better it works. But unfortunately, polystyrene surfaces have a different structural feature than nitrocellulose or PVDF-type membranes. And the basis for the way they operate is the milk does not stick and block as well to the rigid, less complex polystyrene ELISA well surfaces as it does on these membranes that are made to basically non-specifically bind to protein. So you can't just use that. You have to use something that sticks better. Sometimes you need to use something that's a little smaller, but you need to use something more creative that will not only coat the surface, but what you don't want to do is you want to use something so big that it leaves a hole for something smaller to come in. You need to use a small enough blocker material so it literally coats everything and doesn't leave like a passage here and a passage there for something smaller to come in and then give you a bad reaction. So unfortunately, you cannot use non-fat dried milk, though some people do with some success. I do not recommend it. The advances in blocking technology have gone far ahead of where the milk blocker routine has been over the years. So.
Great, thanks, Brian. We also have somebody asking why some blocker stabilizer products give them much lower background signal noise than others they've used.
The blockers will vary by the fact that their composition is variable, and in some cases, when you have smaller molecules or a different type of molecules, whether you're using a synthetic type, detergent type blocker, whether you're using a non-mammalian protein type blocker or a carbohydrate type blocker. We've got all these. Depending on the situation, plate surfaces coated with one or another of these other type compounds will actually resist non-specific binding better than maybe a traditional BSA type blocker. Not that BSA doesn't work, but you've got situations where you got something really sticky and you want to get the assay done. You have to switch around a little bit and see, well, maybe if I coat this plate with something a little different chemically, maybe it's not even a protein, and some of our blockers are non-protein in nature. Sometimes coating with that will not only allow you to stabilize that plate, the components that are on there, but it also will keep the stuff from the sample matrix off. And if you keep that stuff off, then you're not going to have the non-specific binding of the conjugate stuck to the plate when it's not supposed to.
Great, thank you. We have someone else asking here, why do they seem to lose so much of their antigen spike readings after they switched from using cell culture supernatants to plasma samples?
That's the matrix effect villain. They know they got a certain amount in their spikes, and when they spike a sample matrix that's very messy, very complex, like plasma, for instance, what will happen is that stuff will go in there, but the capture antibody in that plate well, in that particular well, will not be able to react as well and capture that analyte as well in that matrix as the corresponding concentration of the analyte in the standard curve well. So the standard curve well will say, oh, you got, again, using the example, you got 50 picograms per mil here. Well, in the sample well, though you don't have this, you're going to look like you got like 20 or 10 picograms per mil in your sample. Now, it's still there. The problem is because the binding kinetics is so poor in these complex matrices, the antibody just hasn't had a chance to bind that same amount of analyte as the corresponding capture antibody can do in the easy, permissive standard curve calibrator type diluent. So it's all a matter of binding efficiency. The lower the binding efficiency, when you read it, you say, oh, I don't have it. Well, it's there, but you didn't capture it because of the interference with the binding kinetics. So for all practical purposes, you lost it, and that's what's going on.
Gotcha. Okay, so we have another person here, Brian, wondering how long should their antigen capture incubation times be if they're running their assay at room temperature?
This can vary a lot with the concentrations that you're trying to measure. Now, if your standard curve, your normal concentration range that you're trying to target is down in the picogram per mil concentration range, okay, that's getting down there a ways. You are going to need a longer capture time, meaning maybe 90 minutes, maybe 2 hours. It's going to, in some cases 3 hours. You're going to take a longer period of time in order to capture a higher percentage of the analyte that's present. Now, if you're looking at something up in the, let's say, growth factor like microgram per mil or something really totally high, because there's so much of the analyte present, by the laws of mass action, it will force itself onto the plate. The capture antibody just has to sit back and, relax, and the antigen comes to it. And so you don't need to incubate for as long. And in those situations, maybe you can get by with something like a half hour, 45 minutes. You can accelerate this process by using a shaker. These are basically vortexers or rotators. And by incorporating a shaker into your incubation steps, you can cut your incubation time from, say, 3 hours down to maybe 2 or maybe an hour and a half. So you get the same results, but the kinetics are accelerated when you shake and rotate the liquids in the plate. So the capture efficiency goes up, and so you can get your assay done in a shorter period of time. I would not trust any assays where you incubate for less than, say, 30 minutes or less, because you want to be sure that you're going to capture everything. And if you're on that steep part of the curve, you don't dare stop that assay unless unless you've diluted it out far enough where you're not going to be in antigen excess. So I would say typically it's at least an hour. Usually it's like 90 minutes, and if you put it on a shaker, you can cut that time by a third or so, and that's at room temp. You can make it go faster at 37, and people have incubators for that. But if you're just putting it in your lab bench and just stick it in the drawer, you're looking at least an hour and a half, maybe 2 hours for the antigen capture to the capture antibody on the plate step. So that's usually your longest step.
All right, thanks, Brian. Well, we have about time for one more question here. Again, like I say, feel free to keep chatting in, guys. We'll respond to everybody else after the webinar if we didn't get a chance to get to you. But for the last question here, how much of a difference does the type of plate or the manufacturer's brand name of ELISA plate really matter when it comes to the amount of work you have to do to finish your assay?
Again, that's another variable situation. If you're just going to run a few samples, have a dozen or a dozen samples over the course of a month's time, and you are not developing a commercial assay, and you're developing an assay where your normal range is real low, like in the picogram per mil concentration, then you can get by with just about any either medium or high binding type ELISA plate. Where things really come into become to matter is when you are trying to develop something that's reproducible, something that's going to be sold commercially, or that's going to be used for long-term accurate assessment of analyte in a particular sample. When you have that situation, you better stick to the more name brands that you can trust, type of plate that are consistent, where basically when you incubate your antibody and absorb your antibody to the plate, they get the same amount of antibody bound on the left side as the right side. They're consistent all the way across. And you want some plate that has the ability to bind lots of protein so that you aren't leaving a lot of your antibody or antigen in your incubation media. In cases like that, then it does matter that you should use a high binding plate, and you should use a good commercial source that's been around for a while, where you can trust their quality control as far as their plate dynamics. So.
Great. Well, that's about all the time that we have for today. Thanks again, Brian. Again, if we were unable to get to your question or your question required a more in-depth technical response on our end, we will follow up with you personally after the webinar. Again, I will also be sending out the webinar recording within the next day or so, so look out for an email from me with the link to that. Our next webinar is scheduled for May 11th. I will be presenting, and it will actually cover tools for fluorescent imaging in cancer research. If you can think of any other questions after the webinar or have ideas for future webinars, please feel free to email me, fill out the Contact Us form on our website, or reach us through social media on Facebook, LinkedIn, or Twitter. So again, thank you so much for joining us, everybody, and have a wonderful day.